Hi, hello everyone again. Hi, hello and welcome. Microbehunter here. I am, as always, trying to get everything set up. So bear with me. Yes, I hear myself. So I hope that sound seems to be working. Okay. It's always a little bit of housekeeping to do every time when I start. And I think we're ready to go. Okay. So hi, it's Saturday again, and yeah, today I would like, as I've already told you in the title, I'd like to look. I've got a box of slides that I bought, but right now, what you're able to see right now in the background, these are two mosquito larvae, still alive in a droplet of water. Okay, you can see the digestive system working, which I happened to collect just about 2 hours ago, because I also a little bit unplanned. I found a water sample that I would like to share with you, so I'm going to do that later as well. But what I would like to do today is I would like to unpack with you this box here. For those of you who are new to this live stream, a short, quick introduction of what we're going to do. First of all, the live stream, I will be, you have the possibility on the side, you have to type questions in the chat, and approximately after 1 hour, I will then answer those questions, or will try to answer those questions. Basically, feel free to post questions. And about what we're going to be doing today here is 2 weeks ago, some of you, thankfully, and again, a big thank you, donated some money. And you still can do that. There are the so-called the stickers and the super chats. You can donate some money, and I used that money to buy a few antique old microscope slides. Here they are in this box, and what I would like to do is I would like to unpack them, and I would like to show them, together. We're going to have a look at them. I did not look at them yet myself. Okay, so it's a little bit, these live streams are also always a little bit improvised, in the sense that sometimes I myself don't even know exactly what to expect. Okay, and so for this reason, I'm going to open it. I mean, I already opened the cardboard just once, just to check that it's the right thing, but I did not actually open it completely. So what I'm going to do is I'm going to simply open it. Okay. And we're going to have a look at those microscope slides. Okay, and if you basically have any microscopy related questions, even if they're not directly on topic, please feel free to ask. Okay, so I will also occasionally look at the live chat here as well. Yeah, and as always, I would like to wish you greetings all around the world, because as always, we have quite an international community, and there are plenty of people joining in from all over the world. Yeah, so that's basically some antique microscope slides that are ordered, using the donations that I received from you. And all those slides, they just that you're able to get the price a little bit. I paid approximately, not quite 100 euros for them. It's around, I don't know, maybe around a little bit more than $100. Okay, and it's not the first time that I've ordered from this shop. This is now the first time that I'm actually unpacking it. Okay, and let's have a look. It's the first time that I'm unpacking this, and the first time that I'm looking at them. And this is basically what I got. Okay, the microscope slides are antique. When I say antique, I'm saying probably around 100 years old. And I've ordered quality microscope slides. So these were actually professionally made slides. And I chose them, first of all, because the contents interested me. Basically, the specimen interested me. But I also like to collect those slides, because, of course, I want to show them to you, obviously. But also because those antique microscope slides generally are of a very high quality. Okay, some of you have already seen a few of my videos that I made before. and actually the quality is surprisingly good. Okay, as a matter of fact, you kind of really feel that whoever made those slides really put in a lot of love. I know it's a weird word to use maybe, but they really put in a lot of love and energy into making those. Okay, so this is basically something that I would like to show you today. So I've got 1, 2, 3, 4, 5, 6, 7, 8 slides I ordered. Okay, and yeah, I already forgot what I ordered. So we're going to take them one by one, and I'm going to put them under the microscope, and maybe then we're going to see if we have a little bit of time. Then I'll also put some of the water samples under the microscope again. But I'm not going to stress myself because of that. I'm removing now the little mosquito larvae again. Okay, let's put them over here. And let's have a look at those slides here. Okay, and in order for me to show you those slides a little bit better, I'm going to put the camera down a little bit, okay, so that you see it a little bit closer. I've got this camera that I can adjust here. Yeah, it's much better because the slide labels are also quite nice to look at. And then I'm going to switch back over to the microscope. Okay, so and yeah, I'm also occasionally also looking over to the chat. Yes, because some people are joining in. So sometimes I'm receiving some info if there are some technical issues as well. Okay, and if there are technical issues, some time ago it was actually interrupted the live stream. Then I'm going to try to restart the live stream again, but I might have to. It might be under a different link. Yeah, those microscope slides. So let's have a look at the first one over here. They're all packaged, as you can see, individually here, yeah, in plastic. Yeah, and my eyes are a little bit bad already, so I actually need proper glasses. What is this? Mounted in Styrax. That's very nice. Okay, it's the mounting medium used. So it's W. Watson and Sons. That's the company name. Okay, London. And over here, what does it say here? Oh god! Oh gosh! Okay. I have a problem reading this handwriting. This was actually printed out when I ordered it, but I don't have the printout now. It looks like a Latinized name. So let's have a look at this here. Okay, what this is. Yeah? Generally, when I touch the slides, I don't touch them like this, but only on the side. And I already mentioned this before, even the glass quality is very high because these are polished edges. Okay, so really nice slides. Let's have a look at this one over here. Okay, so let's put this in under the microscope, and let's have a look what this is. Here is I remember. These are some what appears to be diatoms. So beautiful, very nice. So let's go up with the magnification. I go up with the brightness, refocus. There's 3 of them on here. Ooh, these basically they look like diatom shells. Yeah, that's actually quite interesting. Why only 3 of them? Because back in the 19th century, early 20th century, what people actually have done is they have sometimes actually arranged those beautiful shells on even in patterns on a microscope slide, and evidently they arranged 3 of them here as well. Yeah. So they have evidently picked them out from a larger sample and simply placed them on a microscope slide and then mounted them. Okay, so let's go up a little bit further. So 40 times. And if you kind of are wondering why the background is blue, that is because of the contrasting technique that I'm using. But I can, of course, if it's too dark, I can, of course, switch off over to bright field. So that is the regular view. Very nice. Okay. Yeah. So these are basically some diatoms. They are made of silica. That's the material of the shell. Oh yeah. Yeah. By focusing, we can actually see that there is a certain depth as well. Yep. So you know what I'm going to do is I would like to show you again the slide label. I should have actually printed out the, how do you say, when I ordered it. So that is the slide label again. Triceratium. Yes, okay, Paul just also mentioned this. This seems to be Triceratium favus, yeah? So this is, yeah, that's the binomial name, yeah, of this specimen here. Yeah. Yeah, let's have a quick look at it, and then I would like to go through the others as well. Yeah? By the way, maybe we have also a closer look here. This is the company W. Watson and Sons from London. As a matter of Styrax, it's also quite interesting because you have to have the correct mounting medium in order to see the details of diatom shells properly. Yeah? So I kind of like this additional information. Yeah. So this is actually quite nice. Yeah. So this is how it looks like, and let's put it back again. Okay, I have to switch over to the scope view. And of course, finding it again is a challenge. Yeah, that is the black ring. Here we go. Okay. I'm going to turn on the blue contrast again, and you can change around the colors as you want. You know what I could try? Didn't try that yet. I'm switching over. I'm going to have a look how this looks in phase contrast. I don't know if this is actually... Let's just give it a try. This is now... Yeah, that's now phase contrast. Let's remove this here. That's phase contrast. Yeah. Well, as has been mentioned, the perfection of nature is amazing. I have to totally agree. It's really fascinating. Yeah. So yeah, let's have a look at the next one. Okay, so I'll take this and I'll put it into my slide box of. That's basically how it looks like in. The whole thing, let's turn it down again. It's using the low power objective. As you see this ring here, that is the ring that holds the cover glass in place. Yeah. So let's do the following. Let's move on to the next one. Okay. 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 The question is if we can actually read it under the stereo microscope. Now, I don't know if this is not going to be a little bit too large. So let me quickly check if we can actually see this a little bit better under stereo. So that's now my stereo microscope. And this is how it looks like. Yeah, this is handwritten, of course. Yeah. Okay. Let's move. I'll move on a little bit because we've got still plenty of slides to look at, and I'll just put the slide here on the side. And the next one here is what's the next one over here? Yeah, I'm gonna take it out first, so that it's easier for me to have a look at this. Just a second. I need to find a safe place to put the slide. So, because I've got some liquid standing around here as well. So let's put it over there. So let's go directly under the, or you know what, for Navicula lyra, that's the name of this specimen, mounted in Hyrax. So that is again the mounting medium here. Okay, and let's have a look at this here. So let's have a look at this one over here. Okay. So, also a very nice looking slide. Let's have a look. We have to go up with the magnification quite a bit now. Here they are. Brightness has to go up. Yeah, they're pretty small here as well. Now, these are also diatom shells. And diatom shells actually have been used, can be used, and are used also to test the resolution of the microscope, because they have the shells. Some of them are very intricate, and they have tiny parallel lines. And if you're able to resolve this, then the microscope actually has a very good resolution. Yeah, here we go. And now we also see those tiny lines. So I just want to show this to you here. If you ever happen to also observe diatom shells, if you see those tiny horizontal lines here, okay, sometimes those are used to actually also test the resolution of the microscope, if you're able to see those or not. Yeah? So that is also a nice specimen. Over here, just look what I'm doing here. I'm on the top, okay, because this is my 60 times objective, but there is a ring here that I can turn. And this is for adjustment of the cover glass thickness. So you can also adjust the resolution a little bit like this. If it's too far off, I think that's fine. Yeah. So this is let's have a look here at a slightly lower magnification again, and let's turn off the arrows, because I think there wasn't just something somewhere else, something here. No, that's nothing. It's just a little bit of dirt. Yeah. And it's evident for me that those diatoms were also placed manually on the slide. Yeah. So, because they're pretty much in the center of the specimen slide, so they must have also been placed manually there. Looks nice. Okay, quite happy as well. Yeah. So, yeah, very, very nice. Okay, so let's move on again a little bit. Yeah. So I'm going to remove this here, and yeah, this one over here is a question that I'm going to answer directly, because it's directly related to this specimen over here. What you see over here is these are the empty shells, right? And what you have normally in here is you have the chloroplast. So they are obviously not alive, because they're on a microscope slide, but not only that, it is possible to actually remove the contents of those diatoms to only leave the empty shell. And this way you're actually able to see the patterns much better than if, for example, the cell contents would still be present. So this is something that I think is also quite nice. Before removing the slide, I always go down with the magnification as well, not to crash the objective into the slide. And let's carefully remove it again. And let's have a look at the next one. So again, I need a safe place to put them. Yeah, what is this here? So these were 2 diatom slides. And this here is what is this? So this is evidently from a different company. This is all the diatoms. Okay, but I cannot read this here. And there is nothing written down here. So let's have a look under the stereo microscope, just for better visualization. Here we go. Unfortunately, this is the lowest magnification I can get. Okay. Sounds like hippocampus. Pleurosigma hippocampus. Obviously, it does not have to do anything with the hippocampus of the brain, but these are diatoms, okay, as well. So unfortunately, no additional information here. Yeah, on the lower label. Yeah. So, but let's put it under the microscope again. For those of you who have just joined in the live stream, I bought, thanks to your donations, I've bought 8 antique microscope slides. When I say antique, they are around 100 years old. How do I know that? It is because the company, if you do a little bit of research, it's the when it existed. Here it is. Look at this. Yeah. Okay. Oh, thank you. Thank you. Another donation. Thank you very much. This basically means I'm going to be able to buy some more slides. Okay. So Yeah, and I think this is actually a good example here because the diatoms here, we can actually see that they still have, some of them still have a content, cellular content. Yeah? So diatoms, because they do photosynthesis, obviously they have also chloroplasts. Yeah? I need to, and I'm going to just go bright field now, I think, yeah. And some diatoms actually are not greenish in color, but rather have a little bit brownish because the pigment is a little bit brownish, yeah. And this one over here it seems to be, of course, not manually placed. I've seen that there was a question or a comment of how people were able to place such tiny objects manually. I'm going to explain this later. It is possible. Yeah, so diatom arrangements actually have been made. And indeed, there are some arranged diatom slides that I would also like to buy, but they are very expensive. And those diatoms are then arranged in very nice patterns. This has been done quite frequently at that time, back in the 19th century. This was actually a common thing to do, to arrange those beautiful shapes into beautiful patterns for microscopy. But those slides are expensive. So, but this one actually is also quite a nice one. We see that there are seem to be also some other other species in here as well, but it's generally quite nice. So let's do the following. Let's go up with the magnification again. Okay, very nice. The thing is the following. Just a comment. Those specimens, essentially, you can find them pretty much anywhere. I mean, diatoms are ubiquitous. You find them on many different places. That is not really the reason alone why I like those antique microscope slides. It's simply also the fact that those slides, they do have a certain history. It's not only the specimen itself, but it's also the slide. Thank you very much. Okay, to Pat, another donation here. Thank you very much. Yeah. And sometimes you have to be a little bit patient because, of course, there are auctions, and then sometimes you have to wait a week, and then the price goes up, of course. And those slides I bought, basically, I was able to buy them directly without actually auctioning. Okay, so I'm going to try the next one. So we've had 3 diatom slides. Yeah, because the previous slides that I bought, were not diatom slides, were mostly histological, but I do have some other specimens here as well. So again, let's have a look. This was basically the slide that I looked at right now. Okay, so let's put it away again. I always have to do a little bit of time management, so it's about 23 minutes, which is fine. We're well in time. So the next one, this is from another company, Arthur G. Doherty. He basically, his slides always have multiple colored rings. I already have got several slides here. When you buy those antique slides, then there are many of them, but many of them were old, but essentially were not made to a professional. Well, not a professional is not the right way, but to a very high quality standard. Were just made quickly because the people wanted to analyze something. But the slides that I bought over here actually were made by professional slide makers, who sold them. Focus stem, okay, and stained using iodine green, it looks like. Okay, so this here is a plant. Okay, and as you can see, the color of the ring has multiple colors. This seems to be some kind of a brand thing, because all of the slides from him basically had those multiple colors. So this is now something that I would like to also put under the microscope. So now this is a pretty thick slide. The glass is quite thick. So let's start again with the lowest magnification. I always have to turn off. So, yeah. Okay, let's have a look. Okay, it's a stem cross section. Yeah. These are actually more interesting, or sometimes more interesting, when you get the complete overview and not zoom in too much. So essentially it seems like the outside, the bark seems to be better present here. Let's have a closer look. Maybe we can actually see the cellular structure a little bit better. I have to always refocus. Yeah. But the problem is I don't know so much about this specimen over here, what I got. Okay. That's a little bit unfortunate. Being a stem cross section, we're now looking, here are the cells. We're looking now probably at the, if it happened to be at the epidermis on the outside here. Let's go up a little bit more with the magnification. Okay, now we see the individual cells a little bit better. Yep. I'd have to do a little bit more research on this one over here. But here the cellular contents is still visible. But there seem to be, I don't know, they almost looks a little bit like crystalline, but these are not crystals. But these might be either some preparation artifacts, because there are some gaps in there. So maybe during the cutting, during the microtoming, maybe this is the reason why those cuts started to appear. Yep. So this is an interesting specimen here, because I'm not able to tell you quite a lot about this, what we're able to see here. So what I'd have to do here is to do a little bit more, as I mentioned already, a little bit of more research to make sense of what I'm seeing here. Because also the individual cells here are not quite as easily visible as I would have expected. Because if it's usually plants, the plant cell wall is usually, if you defocus a little bit, you might be able to see them a little bit better. Yep. So this is an interesting one, but maybe not a very pretty one. Okay. Nonetheless, that is essentially something that also sometimes beginning microscopists experience quite a lot. They're seeing something under the microscope. It might look interesting or not, but the problem sometimes is you don't really know all the time what you're looking at, and it's sometimes difficult to make sense. Well, right now we're able to see a little bit some of the cellular structures here. Let me point the arrow, right? You see over here, yeah? Cell wall, cell wall, cell wall, yeah? But for a plant specimen, I have to tell you, it's not quite, the cell walls are not quite as pronounced and as easily, clearly visible as I would have expected. Yeah. So I would say, yeah. Okay. This basically, I've just seen read my notes above. Gary is mentioning something. Yeah. Okay. Iodine green is used for staining chromatin and amyloids. Oh, thank you very much for this. Okay. Chromatin is DNA. So basically, that could be a little bit the reason why it doesn't quite look as expected, because the stain that was used for staining Chromatin. Chromatin is essentially DNA. And iodine, of course, also stains starch, amyloid. Yeah? So interesting. Okay, in any case, I'm going to move on because I still have how many? I still have 4 slides to go. Okay, which I would like to share with you. Again, I quickly show you again the slide. So this was the slide. Okay, with iodine green. So let's move on again to a safe place. This one over here, I'm just going to, it's another plant. This one looks, here it is how it looks like. Okay, it's a cross section of a leaf to show cystoliths. Okay, cystoliths, these must be some structures, maybe some crystals inside the cells. Okay. So that is Unfortunately, I've seen this already several times, but I don't really know what this handwritten label actually means. So let's put the scope again. See where it is. Where is the... I'll probably have to refocus the whole thing. Just a second. Here we go. See, that's what I call a plant cross-section. Okay. Let's go down with the magnification so to get a better overview. Let's remove the prism so that is... I mean, this is a really nice one again, yeah? So, and this is the cross section of a leaf. I think, wow, this is actually a really nice one. Just look at this. Maybe some of you remember when I already looked at leaves before. And I did a small biology lesson as well, where I mentioned that on the top side of the leaf, generally the cells are very regularly arranged. That might be the top side here, and then on the bottom side of the leaf, the cells are arranged much more loosely. It's the so-called the spongy mesophyll, right? And this is what we see here as well. This seems to be here the palisade layer, and this seems to be almost like the upper epidermis over here, because the upper epidermis is usually free of chloroplasts, and this might be the palisade layer, but this upper epidermis is pretty thick here as well. So this is quite a nice specimen, but I will do the following. Let's go up a little bit here, and so this is now in bright field using the natural colors, so to say. Let's do this away again. So, those maybe these are the so-called these little what is it called? Cytoliths. Where is this? I would suppose, Maybe some kind of lith is actually something usually refers to crystal or some kind of a solid structure, and many plants actually have those, and maybe this is actually one of those that it's actually referring to. Either for storage, or in the case of water lilies, they also have those inclusions, and they're actually used simply as waste products even. So this is a very nice example of a very good cross-section, because sometimes, also some months ago, I think I did a comparison between cheap and more expensive microscope slides, and sometimes I've seen some microscope slides where the specimen was cut so thin that, yeah, you couldn't see a lot of details. And this one seems to be cut just in the right thickness. I suppose that it's obviously it's also stained. That's why you see the different parts here stained differently. Those parts here in the center, these are usually always the vascular tissues. Vascular tissues means these are the cells that transport water. Usually the largest cells in here, that's usually called the so-called xylem, which basically are the cells that transport water up to the plant. Yeah? And when we go down with the magnification a little bit, yeah, we should be able to see that, yeah, again, the overall arrangement. And I think it is a very nice one. Let's remove the arrow again. Cystoliths are calcium carbonate stored in cells. Thank you for the comment. So that's, yeah, cross-section of a leaf. Very nice looking. Yeah. This here, again, this bulge here, this huge bulge seems, yeah, to be, again, the tissue that, on the bottom side of the leaf, where basically you have all of the vascular tissue in there for transporting the substances. When you turn a leaf around and you're actually on the bottom side, you're going to see that actually you can see those we call them the leaf veins. Yeah, basically all of those structures that make the network of the leaf that kind of distribute the water. Yeah, also very nice specimen here. As a matter of fact, it's actually one of the better ones where I have a plant leaf cross-section. Yeah. Yeah, it's actually quite nice. And let's just zoom in a little bit much more again, just to get a little bit of a closer view again. And then let's go on with the next slide. Yeah. Yeah. Yep, quite nice. Okay, I'll move on. I shall move on. And where is the desk view again? I got 3 more. What does this mean? Oh gosh, I think I'm going to put this again under the stereo. Shin of poo. That is something that brevis, yeah, whatever. Okay, so yeah, I don't know what this is. You see, I just looked at the slide, looked at whether it's kind of nice looking, and unfortunately I did not read in detail what it is. This is a very thick slide of very high quality. Let's go again to the scope view. Oh yeah, I'm already there. Yeah. Oh, what in the... I've got things here where I don't even know what it is. Okay. In any case, this is pretty nice. I think, you know what I got? That's one. Okay, let's have a look at this, and then maybe I'm going to show you the slide label again, and then maybe some of you are able to figure out what it is. This is quite something. I mean, this... Yeah. The individual cells, I don't know if these are cells. No, I don't know. That's quite something. Let's go up 40 times. Okay, yeah. Interesting. Little bit of a mystery object. Let's go down with the you know what I'm gonna do, because I'm going to now switch completely. I'm going to show. Let's have a look at this under the stereo again. Just a second. Stereo. Here, this is under the stereo microscope. Because maybe we're able to get a better view of this. I mean, this is definitely too small for any plant cross section. It seems to, I don't know, is it some kind of also a shell of a diatom? I don't know. Yeah. Doesn't look so good under the stereo microscope, but at least we're getting an overview. And just a second, here's the camera. Yeah, no, it's better under the compound microscope. Okay. So that is the label. Oh, that's not an H. That's a spine. Oh, spine. I just show you. I thought this is an H here, like shine, but this seems to be maybe an old way of writing a P. Ah, I remember. Yeah, thank you. It's also, yes, this is the cross section. Now I remember when I bought it. Yes, that's correct. It's a sea urchin spine cross section. Yes, now I remember. Okay, when I bought this. The spine of, sea urchins, they have those spines, and this is the cross section of one of those. How in the world did they manage to cut that? Because usually those spines are pretty hard. But let's put it again under the compound. See, that's also one of the nice things here about the community, that while I'm doing this, some of you are actually researching this. And also some of you have already specific knowledge. Yeah, so that is, quite a pretty thing here. So these, that's the cross-section of a spine of a sea urchin. Ah, of course. Let me see. I don't have it here. No, it's not here. I have also spines of sea urchins, but not here right now. But not in cross-section. Yeah. So let's have a look at the next one. I see that some of you are asking questions. Don't worry, I will try to answer them, okay? But I'll do this later, okay? Please, don't feel disappointed if I just do not respond to your questions, because there has been the request that I basically answer them in one bunch towards the end. So 2 more to go. Here they are. Let's have a look here. This here is Helianthus annulus. It's the longitudinal section of a stem, so it's another plant. Stained picrocarmine in balsam and benzol. So down here again, how it was prepared. And this is a longitudinal section, LS, of a stem of a plant. So let's have a look here. And again, the scope view. Ah, yes, very nice. You can see the plant cells again, different tissues. But maybe I'm going to go down again with the magnification, simply to let's remove. It is. I do not have. So the colors that you see here are now basically because of the stain, because I did not add any. No, just a second. There are no. Yeah, so the blue and the yellow basically are. This is interesting. Maybe it's somehow. Uh, Actually, I removed the polarization unit, so I don't know why it still seems to polarize here a little bit. So that's okay. That's now in bright field. Yep, nice. Interesting as well. I would have expected to see some xylem tissue here as well. That's, but yeah. So what we see here is what I suppose is the following: is you see over here on the side, you see of course the individual cells, and those and those long things usually always are for transporting substances up and down the plant. Yeah. So this is actually. If this happens to be xylem, then this basically means that the cells have lost their basically the dividing walls to make one continuous unit so that the water is able to flow continuously. Not normally, but in many plants, what you see is you see that the xylem is also reinforced, and then you see those spiral-shaped structures. This is actually something that I would have expected here as well. Yeah, but those parallel lines are usually always there for transporting substances. Yeah? Yeah. I wonder. Yeah. Here again. Yep. Also quite interesting to see the different types of tissues. Yeah. Yeah, the longitudinal section is of course quite different than from, for example, if you were to cut the stem transversally, then you would see again the different cells and the different arrangement of the cells. And here you actually see that you see it basically from the side, and how the different tissues kind of are alternating. Yeah, interesting. Let's go all the way to the end here. Okay. Patterns of nature. That's all I can say here. So let's move on. Again, last one. What time is it? Because I do want to show you before I start to answer some of your questions. I do want to maybe have a short look at also at the water sample that I collected today. And the last one over here is this one. Yeah, it's again one from W. Watson. Oh, yeah. Starax. So let's put it again under the stereo. Arachnoidiscus ornatus. Arachnoidiscus. Arachnia is actually spider. Yeah. Yeah, this is how it looks like. There seems to be, it seems to be again quite a small specimen. Scope view. Here it is. Yeah, there's three again of them. Yeah, again diatoms. So these are evidently also placed into this arrangement. And let's have a look at one of them again using 60 times. Again, nice patterns of nature. Usually, these round diatom shells are marine diatoms. So if you see, I mean, nature is very diverse. So So maybe there are also some round ones in fresh water, but generally there are plenty of round ones in marine, in salt water. So again, a diatom shell here. Also nice specimen. And I'm again very happy that the slides survived such a long time. Yep, that's how it looks like. If you have also, I mean, diatoms are very easy to find, and generally they also look quite nice. As you can see here, if you play around, also in this case, I'm now using polarized light to give the colors. But if you try oblique illumination, for example, there are certain filters or dark field. Generally, diatoms look very nice. Yeah. Yeah. So quite beautiful again. In natural samples, what you're often going to find is you're sometimes going to find fragments of those. Yeah? Not the complete ones. So that's actually one of the ways that I know that they have been placed, because they have been selected and then placed together, 3 of them in the center of the slide. If this were basically a sample directly taken from nature, there would be many more contaminants around it, fragments of diatoms, and so on. So actually, this basically, there's quite a bit of energy was needed, quite a bit of a time investment as well, is to make those slides than at that time. Because if you take simply some sand containing diatoms and you put it on the slide, you're also going to find them, obviously, but a lot of other things as well. And here, because these are only the diatoms and placed nicely next to each other, is actually shows that the person who made the slide actually arranged this. Okay, yeah. What I'm going to do now is I'm going to move on. So thank you again for your donations 2 weeks ago. Because of that, I was able to buy those slides. I just want to give you a last overview. I mean, what I would like to do in any case is I will also make occasionally YouTube shorts or also separate videos using those slides. Now I have a problem actually picking them up. Okay. But already the slides themselves, you know what, I'm going to move this up a little bit more. Already the slides themselves look quite pretty. Yeah. So that's basically the things that we looked at right now. Okay. And then I have to put them into my slide box. Oh. Picking up. Yeah. Oh, okay, here we go. And there's another thing that I just wanted to show you here, especially with this one slide here. This slide here, you might not be able to see it, but it's almost, I don't know, extremely thick. Yeah. So you just by kind of simply holding those slides, you already feel that they've got a pretty good quality. Yeah. So okay, so this is basically this was basically part 1, so to say, and I would like to move on to part 2 for a couple of minutes, and then I would like to respond to some of your questions. Okay, so for the next one, I'm going to make some slides now, and for this reason, I really have to make sure that those slides are put into the box. Just give me 10 seconds. Okay, here we go. I'm just going to put them quickly into the box. So up. Yeah, here are my other slides that I already have, also antique. I'm just going to put them in here for right now. And this was a first look, but I'm going to, as I already mentioned, make separate videos. But maybe I'm going to make a separate diatom video where I'm going to show them to you in a little bit more detail, maybe a little bit with more information after I researched a little bit more. Okay. Ah, yeah, I see already that the questions are here. Thank you very much. Okay, I got your questions here. But I will only answer them in a couple of minutes because I would like to first have a look at another, specimen that I collected today. Put them away so that they don't fall over. So, where is this? Yeah, I'm going to change topics now, okay? And I'm going to quickly show you something, a very short video. This is basically the place that I visited 2 hours ago, okay? This was in a garden of a relative. And this is basically some rainwater that was collected for watering the plants. And you can already see that I've got some plastic containers here, and I collected some algae and some water. And I just wanted to look at the water now, okay? And I just wanted to show you I've done a little preparation here as well, because if you look very carefully, I've collected a tube of water and I centrifuged it for a couple of minutes. So I've been spinning it very quickly, and do you see this green dot down here, this tiny green dot? That is the pellet. These were basically the suspended algae in so much, I don't know, 10 milliliters of water. Not a lot. The water looks significantly greener than it actually is, because the greenness is not so, it's a little bit from algae that are kind of floating in the water, but it's mostly green because of algae growing on the sides of this this container. Yeah. So this was actually surprising to me a little bit that there were not more algae in there. And I'm just going to show this to you here again. Where it says, no, that's the wrong one. Here it is. Do you see this green? Yeah, that basically, this green thing here, that is the pellet. And I would like to put this under the microscope to see which type of algae are present. And I really don't know if there's anything interesting in here or not. So that's why I would like to try this out. So, and in order to do that, so I've been centrifuging this for approximately, 10 minutes or so. I've removed all of the excess water. So for those of you who don't know about centrifuging, I put the water in here, and I've got a centrifuge which was spinning it very quickly. So all solid particles that are denser than the water kind of accumulated down here. Usually the pellet is significantly larger, but this is going to be enough. This is going to be enough for microscopy. And the excess water I then, of course, carefully removed. So what I have to do now is I have to take a microscope slide, obviously, and I have to resuspend the pellet. So I'm going all the way down. There is still a small drop of water left. And I'm pulling it in and drawing it in and out to resuspend the pellet. And there is... it's a small amount really, that's not a lot, but it's, and before I do that, just a second, let me quickly wipe the slide again, because to me, no, but it's clean enough. So, and let's put it in here. We can see it's, or at least I can see that it's green. So there are algae present, but I don't know which ones, and I don't know if there's anything interesting in here. And I'm going to quickly wipe the cover glass again. And let's have a look. Okay, it's a very small amount, and let's have a look. Scope view. Again. Oh, okay. I think we need to definitely go up with the... A bit of movement as well. Yep. This is how it looks like. and 40 times. Somehow those cells seem to have survived the centrifugation process as well. And we see an assortment of different algae. Don't know what those moving ones are, the moving cells are. But we see that centrifugation really is a very good way of concentrating the specimen. Yep. There is this little Yeah, so in other words, what we see here is obviously those that are green. Yeah, these are of course doing photosynthesis, and those smaller ones that are moving around, either small ciliates or flagellates. Probably not bacteria so much, the larger ones here, because bacteria, they would be too large for bacteria. I can try the following. I can actually go into phase contrast. Have a look at phase contrast again. Yeah. Doesn't look so nice. I think I'm just going to stay with the other one. Yeah, so that is basically what I'm able to see. 7, let me put this here. Okay, there's now 60 times. Close this here a little bit. And now I probably would have to take out my identification book and try to estimate what these things could be. Yeah. Again, the biodiversity is so high that, if you are kind of interested, I know that, look, there is a slightly larger one over here. Normally, it is like this, that these cells that you see here are so-called planktonic. Planktonic means that they're floating in water. If you've seen already in other videos, the larger ones, like, for example, I don't know, Stentor, Vorticella, Rotifers, and so on, they like to adhere to a surface. Right, so that means you have to take some material from the surface, scratch some material off from the surface, and then you're able to find a completely different organisms. But these are organisms that were floating freely inside the body of water. Yeah, so they are quite different than the ones that you are able to collect from the surface. Sometimes those round algae sometimes belong to the chlorella, but generally I do not recommend trying to identifying organisms with a microscope also has in some cases limitations, because sometimes you're not able to see all of the necessary structural details. Look at this. This is also, that's a little interesting. I'll show you. Do you see this long thing here? And there are some algae associated with that. It almost looks like a worm, but honestly, for a worm, that would kind of surprise me. For a worm, this would be extremely thin, way too thin. It could be, I don't know, I'm also hypothesizing now, some cyanobacteria, are also able to move. But then again, biology is so diverse. Why not have a worm that is that thin? But that honestly would be one of the thinnest nematode worms that I would have ever seen. Because just compare this to the cell size that you have here. These are those algae that you see are individual cells. And a nematode, if this were a nematode worm, then which is a multicellular animal, then the cells must be yet smaller. Which is possible, yeah. That's quite nice, huh? So. Look how the cells are aggregated around it. Yeah, and so that's basically what I usually like to do. I put a sample under the microscope and simply observe a little bit of yeah, what's visible. Huh. Yep. Okay, people. You know what I'm gonna do now is I'm going to yeah, I think I'm just going to leave this now in the background, but I'm just going to also try to address some of your questions as promised. Okay. Or no, you know what? I'm just going to show you the desk here, and yeah, I got a couple of comments and questions here, and yeah. I hope that I'm able to respond to most of them. Some of them might be in reference to some of the things that I've already shown you, so it might be a little difficult for me to, yeah, address it and clarify it, but we're going to see. I may have missed it, but do you have a case of some kind of a display, the antique slides, and enjoy them when not in use? Ah, yes, that's an interesting one. What I normally do is, and I know that this is not the point of your question, because this is not a display case, right? I have a slide box, yeah, and this is basically how my slide box looks like. I've got all of my how do you say, antique slides, and here look, some of them are actually paper covered. Yeah. This one is so thick down here that I even had problems putting it in here. Yeah. So that is essentially simply a slide box that I have. When I'm going to sort through the whole thing, and then I'm going to make an index over here. But of course, it would be kind of nice to have some kind of a way to have this also visible when they're not in use, right? And this, maybe in a safe way, those slide boxes, if you drop them, you don't want to think about the possibility, right? So, and yeah, so kind of having a safe way of actually displaying it would be nice, but I don't have one. Would be also quite an interesting thing. Maybe some kind of a behind glass, maybe some a way to kind of display it. Morina, we could read the name under the loop or stereo. It seems like a nice collection. Congrats with your purchase, Microbehunter. Thanks for sharing the fun, and thank you for your comment. Okay. What are those internal structures? I think this was in reference to those structures from the diatom shell. Those diatom shells are made of silica, which is chemically similar to glass. Many shells, they're called frustrules. Many shells are made of calcium carbonate, but those of diatoms are made of a completely different material. Yeah? And yeah, you have those internal structures as well. And inside those diatom shells, you have actually the cellular contents, the nucleus and the chloroplast and all of these other cell organelles. Please take the money you buy and buy berry jam. Yours worried me in the last videos. Okay, some information here is I made some videos about raspberry jam. I think 2 or 3 videos about those, and 2 videos, and I found some interesting structures inside the raspberry jam that looked like worms, but actually it was called plant trichomes. So these were basically not worms inside the jam. So this is kind of referencing this. Yeah, they didn't change anything, any particular difference between this one and nowadays. So I suppose maybe are you referring to the different slides? After any particular differences between, maybe I just assume the antique slides and the modern slides. Well, in short, the modern slides, you can have a huge range in different quality. Modern microscope slides, when you buy them also from good companies, can also be fairly expensive, because sometimes the specimens are very rare. Sometimes modern microscope slides are made specifically for medical instruction. Okay, so for example, when doctors or med students basically will look at them and will study them, because they have to be able to recognize different structures, different tissues under the microscope, right? And for this reason, I've seen that some modern microscope slides can be quite good in quality, but generally very often they do not spend so much time and energy in making the slide itself look very beautiful, right? It's I've also seen some very cheap microscope slides, And when I say cheap, then you can actually see that it's cheap, because sometimes when you look at those slides under the microscope, you're going to see that there's a lot of dirt, a lot of dust. Okay, so that is usually the problem. Yeah? So, so a protein-based stain. Okay, yeah, that's about the stain. Yeah, yeah. So that's basically yeah. This was this this iodine stain. Which yeah. Would say the previous might be brown algae. I don't know to what this is now referencing. There are also, they're not only green algae, but of course they're also brown algae. And this one, I don't know now if what this refers to, maybe to the leaf. Yeah, so these were the cystoliths, are calcium carbonate stored in the cells. Yeah, so these are indeed the little tiny crystal-like stones that we've seen in one of those samples that were basically or can be found inside the cells. I have a question. Do red leaves have red chlorophyll? Well, chlorophyll actually is green, but there are also other photosynthetic pigments around that are not green. Yeah. For example, it can go into the brownish color, reddish color. So, for example, water plants that are further down, in greater depths, they have a more reddish color. Right? So there are a variety of different photosynthetic pigments around. Who are the suppliers of the slides? So basically the slides that I showed you today I bought over eBay. Okay, and there are some people out there who actually have bought them and who are selling them. And the price range can be quite large. But generally, it depends a lot on also the rarity of the slide, and also a little bit on the specimen itself. So I found, for example, that those beautiful arranged diatom slides are quite expensive. Yeah. Please try the spine in dark field. Ooh, okay, okay. See. Yeah, I don't know if this is such a good possibility, because the spine is fairly large and I don't know which one it is now. Oh, gee. Is this? I forgot. I have to tell you, I forgot now what the spine actually, which one is. Otherwise, I'll try to make it into a separate video. But you know what I'll do is I'll try to answer maybe the questions first. But the spine in dark field, I'm kind of think it might not be very well visible. I forgot honestly which one it was. Okay, I don't want to go through all of the slides again. Okay, yeah. I thought sea urchins are invertebrates. They are. What actually is the spine? Oh, okay. I get it. It's a word confusion. It is correct that sea urchins are invertebrates. They do not have a spinal cord, okay, like a backbone. But those spines are, I don't have a pen now. Gee, I don't, you want a pen? No. Basically, they have spikes. Yeah, going out like yeah. So, and those spikes are called the spines. And when this was actually a cross section of one of those spines. Okay, so the word spine has a double meaning. Yeah, so that's the reason when you step on a sea urchin, it kind of hurts, obviously, because of those spines that kind of might hurt you. Yeah. Helianthus and sunflower. Okay, thank you very much. This clarifies. Yeah. Is a species of sea urchin. Okay. Now we know it. Okay. Sphaerocinus granularis. Let me see. Let me see if I can find this. No. Helianthus. Helianthus. That's the. This was basically the stem. This was the sunflower. So that's not the one. Spine. Here it is. Okay. That's okay. I found it. Where is the scope view here? That is again the sea urchin. let me see. Where is it? Here it is. And the request was to look at this in dark field. I only have one dark field setting, and that is the one with the 4 times. And evidently I'm not able to see anything right now. Evidently, let me see. Evidently the difference in refractive index is not large enough for me to see it. Seriously. Okay. If I open this too much. No. Nope. So this is actually a sign that, let's try it like this. Ah! Wrong setting. See? I forgot the condenser. So, let me refocus this. Here we go. It actually looks surprisingly nice. It's too much. So that is now the sea urchin spine in cross section in dark field as requested. Okay. Yeah. So that's by the way, dark field is actually quite easy to achieve. If you've got a microscope with a condenser, then making up dark field filters is quite easy to do. So let's move on. Okay. Yeah. So that was basically the one that I showed you before. That's also good. See, that's again a disadvantage because if had I read those questions before, then I could have yeah. So that here is the sunflower. Okay. So remember, this one over here was the stem of a sunflower, square scope. Now we have to remember this one over here? Okay, that's a sunflower. Okay, seen from the side. Unfortunately, I don't have the one. Maybe that's actually, you know what, good topic for video. Cross section and longitudinal section. Okay, it looks very different. Sunflower. And the desk. Okay, so what is the stain used for the Helianthus annulus? Okay, that's the one that I have over here. So let's have a look what they did to actually prepare this. Okay, so that is the sunflower. It's stained with carmine. Picro, I don't know, but carmine is actually a very common stain. It's also a food coloring. And carminoacetate is also used for staining the nuclei for DNA. And the second line in balsam, that's Canada balsam. Canada balsam was a very common mounting medium back in the day. Difficult to use, but very long-term stability. When it says balsam, it always refers to so-called Canada balsam. It's difficult to read again. So let's do again the stereo view. Okay. Carmine is the red stain, and Canada balsam and benzol. And that's the mounting medium used. That is the sunflower stem. Okay. LS stands for longitudinal section. That is the person who made the slide. Maybe he did not make it himself personally, but usually, well, at that time, because there were so many of them made, I suppose that he had his own company, and his own employees that actually made the slides for him. Yeah. So, but very nice. And next one, where is the desk view? Okay, I read about an easy experiment where you suspend one or more slides in pond water or maybe a mini aquarium and leave it there for a couple of days. And this is in order to create a so-called a biofilm. Did you do this and how did you do it? I did not do it yet, but intended to do it. But thank you for reminding me. This is, you do that in order to create a so-called a biofilm. It's very easy to do, and I just want to show you. I don't know if it's very good. I don't have a good specimen here, but sometimes let me explain it to you. When you have a home aquarium, or when you have a glass jar with pond water, and when you leave it for a long time, and if you do not have any snails present, because the snails eat away the algae, right? Then you can see that the inside glass of the aquarium or the glass jar, algae will start to grow and it will become green. And of course you can try to scratch off those algae, put it on a microscope slide, but the problem when you do that is that of course you end up destroying the natural structure of the biofilm. So what you can do is you can actually take a microscope slide and you simply place it into pond water for a couple of weeks maybe, and then you have essentially algae hopefully growing on the glass surface itself, right? And then what you can then do is you can take this and directly put it under the microscope without actually damaging the natural composition of the biofilm. Yeah? So this is actually something that I've not done yet. But it's something that I observe every day when you, for example, have a jar of glass water standing around. I just want to show you something else. Look, just to illustrate this. I like to use those water bottles, because I do not have running water. So I've been using this now for quite some time. But if you look carefully, you're actually going to see that it's difficult for you to see now, but actually on the inside there is a discoloration. It looks a little bit greenish on the inside. So essentially when you let it stand for some weeks or months even, then algae will start to grow on the inside. So this is also a biofilm that has been forming naturally over time. And you can actually do that also deliberately by placing a glass slide directly into some pond water for a couple of weeks, and then you are able to grow directly on the slide the specimen that you would like to observe. So what I would like to do now is so there's a request here at the end, basically to read the questions after each small topic. That would also be a possibility, that I basically break up there's a question here, read the questions after each small topic. That way they can refer to that part only. Okay, I get it. Also a possibility. So because I've been talking about several topics, like for example the microscope slides and then the pond water and so on. I'm still trying to find the best way of actually going through the whole thing. In the past, it was like this, that I tried to answer the questions right away. It was a little bit one extreme. And then sometimes I got very sidetracked, and then people requested that I kind of block the questions together a little bit, but maybe it's a little bit too much together in once. What I have at the end, what time is it? You know what I'm going to do now at the very end? Remember this one here, right? Do you see that there is some stuff floating on the surface of the water? Okay, I've got it here. And that's the last thing that I would like to put under the microscope. Yeah, I've got some of this stuff here, right? So I'm going to put some of this here also under the microscope, and maybe we're also able to find something here. And you can already see here that the water itself looks relatively clear. So that's why the pellet was quite small. So, and then it's been again 1.5 hours, and I'm going to call it again quits. And we go all the way down and take us. I've got some here. Okay, and this is basically also where I got the mosquito larvae from. Yeah, which I showed you right at the beginning, which I put somewhere and I don't know where. Yeah. Oh, where are they? I've got such a big mess on my table that I misplaced them. So and the cover glass goes on top. Again, I'm going to quickly dry wipe them. Okay. Yep. And let's have a look at this. So, yeah, of course I have to remove the antique slide first. I do not want to have any wet material next to the antique slides, next to any permanent slides, obviously. Oh yeah. So, and let's have a look at this one here. Again, also the first time that I'm looking at this, and let's have a look. I expected a little bit more movement, actually. Homogeneous algae. Sometimes in this floating scum, pond scum that you see, sometimes you see a lot of diatoms, but not in this case. Here the movement because of capillary action. Oh yeah, there's something here in the middle. Those fine lines that you see here sometimes can be cyanobacteria. Most likely they are. But otherwise, no. Well, a little bit of movement here. Okay. That's probably some of those that we've already seen before when I centrifuged the sample. Yeah, but otherwise, you get the idea. Samples are sometimes quite different. No paramecia, no microanimals like rotifers. What is this here? I don't know. Could be rotifer. Yep, so yeah. And those thin lines that you see here, most likely cyanobacteria again, or other bacteria. But often cyanobacteria, they grow in long filaments, but they're also pretty thin here already. So it could be maybe, yeah, cyanobacteria often grow in filaments or can grow in filaments, so maybe these are some. Okay, what I'm going to do now is I'm going to, I'd have to look around a little bit more. Maybe if you spend a little more time, you're going to be a little more lucky and maybe you're able to find some more interesting things here. But then that's also part of the hobby a little bit is to let yourself be surprised. What is this? This looks like a rotifer. Okay, here we go. Finally found one. Yeah. Okay. What I think I'm going to do now is after 1.5 hours, I'm going to call it quits again. Okay. Thank you that you've joined me again. Thank you also for your support, for your financial support. I'm going to use this money again to buy a new microscope slides to show you, or other things that I would like to share with you. Hope to see you again next week. If you have any requests or any suggestions on what I could put under the microscope, please do tell me. Sometimes I make up my mind in a very short notice. I mean, for example, today in the morning, as it is, I'm just going to show you the antique microscope slides. But then later on in the afternoon, just a short time before I started the live stream, I was able to collect the water sample. That's what we're looking at right now. So you see that the whole live stream is a little bit spontaneous, a little bit improvised, and this kind of a little bit part of the format that I would like to discover the things a little bit together with you. Definitely a rotifer. Okay, but if you have any specific requests or wishes that you would like me to put under the microscope, then please feel free to ask. Okay, thank you again. Got another. Yeah. another support. Thank you very much. And I'm going to, just an organizational announcement as well, just because some of you are maybe have also checked my website and the forum. Over the last couple of days, I've been struggling and still not fully resolved. The forum sometimes says that there are too many requests and it displays an error message. And also my website sometimes displays an error message. YouTube is fine, okay? So YouTube is not affected, obviously, but my website has some issues, and I'm trying to fix it. But please be patient, and if the forum or the website continue to display errors, you just have to click the refresh button a few times, and then eventually it will work. But I mean, I'm in contact with my hosting provider, but we were not able to locate the problem yet. It's just some organizational matters here. I'm going to now call it. I'm just going to last couple of questions. Is it possible, or can you do streams in the morning for some parts in the world? It's easier to watch. Difficult for me, unfortunately. Okay. Occasionally, I want to tell you as well. Occasionally, I'm not able to do a live stream in. On a Saturday, and I will try to do it on Sunday, but usually it will be also at the same time. It's otherwise relatively difficult for me to. Maybe I'll try to do in the future some maybe shorter versions in the morning. I'll have to see how the whole thing develops a little bit. But in any case, you are able to watch everything later on as well. So all of the live streams will stay online. And because 1.5 hours of a live stream can be a little bit long sometimes, the advantage later on is that you can also double the speed. In YouTube, you can adjust the speed a little bit, and then it also takes less time. Okay, so I wish you all the best. Thank you very much for having joined me again. If for whatever reason it doesn't work out for me to do it on a Saturday, please do read the community posts. But generally, and otherwise, see you around next week again. Happy micro hunting and all the best. Bye bye.